हॅलो गाईज, गुड इव्हनिंग. आलेत का सगळे? चला, जे जे तुमचे मित्र कंपनी ॲब्सेंट आहेत बोलवा त्यांना पटकन. Today we are going to discuss about the enzymes. Okay, how they looks like, what are the enzymes, why they have incredible importance in our biology. So it's already a 4 minute late, guys, and it's a long topic. Okay, so without waiting for the latecomers, let's start with the enzymes. So good evening and welcome to Unacademy. This is Abhijit, and today uh, we are going to see the enzymes, how they functions and how they nomenclature also. Okay. So the name of enzyme, the function of enzyme, the reaction which is carried out by the enzymes. Also we are going to discuss. Okay. So welcome to Crack Maharashtra CET Maharashtra examination. But before starting the lecture, it is necessary to introduce myself. So this is Abhijit Madkar. I have 6 years of experience in biology in 11th and 12th standard. I also cracked a NET CSIR examination in December 2016 with All India rank 60 from the life science. So we call it a specialization in life science. Okay, because exam I conducted was the life science. So you are here on India's largest platform, so you can meet a different educator on this platform. But it is necessary to download this app in your mobile or in your cell phone. It is available on App Store. Please download this app and then you can use it more, and uh, you can meet our different educators also. So for your daily updates, it is necessary to join this group. This group is on Telegram. If you don't have this Telegram app, first of all please download it from your app store and then copy this link in the search box, so you can join us our channel after hitting join on the bottom of the screen. Okay, so this is the subscription. If you take a subscription of Unacademy, so you are able to use it unlimitedly. What is meant by unlimited? So here if you see, the subscription for 12 month is 16,500, but if you use this code Abhijit in a capital with a subscription, then you will have a discount in the fees and it will only 14,850, and for 24 month it is 21,600. Okay, so what is extra in a subscription is you can uh, you can take a lectures or the personal education from your favorite educator. There is also session for the MCQs. There is also session for the doubt solving. Okay, so doubt solving session is extra in this subscription. You can use this app unlimitedly with a single subscription. Okay, so it is necessary to take a subscription for the unlimited app and the different educator, or to meet your favorite educator. So guys, let's start with the enzymes. Okay, so you can see this flower is opening itself. It is the effect of an enzyme. Okay, so in nature what is happening in living beings is everything which is a reflection of enzymes. Enzymes are everywhere. They conduct each and every reaction in our system, I mean in a life. Okay, so from organism which is a bacteria, a single cell organism, or you can say a tiny life on earth, to the gigantic organisms which are the largest in a shape. You can take a blue whale or the elephant also. So these type of organisms are also living on these enzymes, okay, because each and every reaction inside our cell is catalyzed by enzymes. So we call it a catalyst of biology or simply a biocatalyst. Okay, so what are the enzymes? Enzymes are the biocatalyst. Each and every reaction in our body is catalyzed by enzymes. So let's see where they are. They are everywhere in living beings. Okay. So thousands of reaction per second or the lakhs of reaction you can say per second are running inside our cell, and all reactions are conducted by enzymes. Enzyme conduct it. Okay. So all type of reactions are conducted by enzymes. If you see the speed of reaction, it is thousands of, or you can say 10 raised to 6, 10 raised to 7, 10 raised to 10, 10 raised to 14 a fold more than the without enzyme or without catalyst. Okay, so a life without enzyme you can't predict it. Okay, or you can't see there may be a life without enzyme. Okay, because if you see the reactions, they are very slow in nature. But when you encounter enzyme, you can see this reaction which take 1 year or 2 year or 10 year in a nature, which can be conducted by enzyme in a fraction of second. So enzymes are necessary for our normal growth. Okay, if you see the lifespan of a human or any organism, it is a small. But if you see the phases which are in the lifespan of organism are more, and to require the fulfillment of these pages, we require a speed of reaction, and that speed of reaction only achieved by the enzymes. So how many reaction per second? So there are a thousand, lakhs, and crore of reactions per second the enzyme can conduct. All cellular reactions are mediated by enzymes. That means each and every reaction inside of cell conducted by, or you can say catalyzed by enzymes. So what is accident with enzyme? So this is a scientist Edward Buckner who first encountered the enzyme. So this German scientist was working with a yeast extract, and he suddenly observed that extract which is of yeast. That means what is meant by extract? When yeast is killed, or the lysis of yeast. Lysis word is generally used in a biology for the for example, if you break the cell, you can say a cell is lysed. Okay, so lysis is used for the breaking of a cell. When the scientists break a cell of yeast, okay, yeast means you can say baker's yeast, or these are the organisms from a fungus group. Okay, yeast is a fungus. It is unicellular and from fungi kingdom. So the extract of yeast can ferment. Okay, that means this cell is not integrated now. This disintegrated cell also shows a reaction which results in a fermentation. Okay, and that time the scientists conclude that there is something which is going to conduct the reaction. So he just name it like inside of a yeast. Okay, so who is conducting the reaction? A unknown thing which is inside of yeast, and in the language of that scientist he written that EL means inside and zyme means yeast. Okay, so yeast can be written as a zyme. Okay, inside of yeast means enzyme. So we use this term as it is. from the scientists till today. Okay, so enzymes means, it doesn't mean that they are only present in yeast, but when they first encountered this Edward, that time he was working with a yeast. That's why he named them like a uh, N and zyme. That means enzymes, which are which are the part of a yeast. So this is a scientist which first encountered the enzyme. So there are two main types of enzyme, which first is a endoenzymes. What is meant by endoenzymes? The endoenzymes means they act in an environment where they synthesized. That means if a particular enzyme, I am taking an example, for example, let's take a mitochondria. Okay, so this is mitochondria. I am going to zoom it. So we generally draw mitochondria like this because it is a two membrane system. Okay, so mitochondria is two membrane system, and inside the mitochondria, for example, this is an enzyme of mitochondria, or we can say this is an enzyme of mitochondria inside the all of inner membrane. Okay, so this inner membrane also contain electron transport chain where the enzymes are present. These enzymes, or these enzymes which is inside of the mitochondria or in the matrix of a mitochondria, they will never conduct any reaction out of the mitochondria. Okay, so these enzymes, endoenzymes, are only made to act or to conduct the reaction inside the environment where they are synthesized. Okay, so they will work in only that enzyme where they are synthesized. For example, mitochondrial enzymes are there, uh, chloroplast enzymes are there. These are the enzymes which are the example of endo enzymes, and there is also exoenzymes. Okay, so what we discussed in a last slide, that is accident with the enzyme, was the exoenzyme, because outside of the cell, out of the cell, they can be conduct a reaction. So we can say they are the exoenzymes. Exoenzymes means they can act or they can catalyze the reaction outside of the environment where they are synthesized. These enzymes are released by the cells like a fungi. Okay, so what is fungi? So let's see, for example, this is a fungi. So this fungi, when they want to grow, they just release the enzyme into the environment or the vicinity of the cell. Okay, that time this enzyme digests, and it digestion or the direct final product can be absorbed by a cell. Okay, so this type of enzymes are called as exoenzymes. They can act outside of the cell, or they can catalyze the reaction out of the environment where they are synthesized. Okay, so only fungus is there which can digest its food outside the cell and then absorb. Okay, most of the time we use a word saprophytic in a nature. That means it is a mode of nutrition. Saprophyte is a mode of nutrition, and this is only concerned with the fungi. Okay, so what are the enzymes? Enzymes are proteins. So the first thing, what are the enzymes? Enzymes are a protein. That means enzymes are made up of protein. Made up of protein. And we know how proteins are made up. Proteins are the a chain of amino acid. So you can say enzymes are the made up of amino acids. Okay, but not all enzymes are only proteins. Sometimes they require non-proteinaceous part to activate. Okay, so all enzymes in a nature are protein, but all protein are not enzyme. What it means? All enzymes are protein, but all proteins are not enzymes. This is a sentence most of the time you heard from your teacher or the friends. but you most of people will not get it. What it means? It means the enzymes are made up of protein. That means all enzymes are protein, but there are different type of proteins. Some are act as a transporter, some are globular, they are function for different work, some are the structurals. Okay, so this all type of protein will not going to conduct the reaction. So this type of protein we cannot say these are the enzymes. Only proteins which are taking part in a catalysis of reaction, in a catalysis of reaction is enzyme. Okay, so there is again exception for this term. Okay, because few RNA also shows a catalytic activity. RNA, A capital A, catalytic activity. That means we had a few RNA in our body which is involved in the reaction, or they are act as a catalyte or catalyst in the reaction. For example, your ribosomal RNA. Okay, so ribosomal RNA helps in making of protein synthesis. That time they are able to conduct the reaction or they catalyze the reaction. That's why only RNA which shows a catalytic activity is exception for this sentence. Otherwise this sentence is true. Okay, so there is only single exception, that is RNA which are going to conduct the reactions. Okay, so there are conjugated enzymes also. So what is meant by conjugated enzymes? These are the non-proteinaceous group attached with a protein, and these groups are called as prosthetic group. That means, for example, this is your enzyme. Okay, for example, this is your enzyme which is going to take part inside of the reaction. Okay, so for example, this is enzyme and which is going to take part in a reaction. That time it requires one extra molecule here, otherwise it will not work. So this molecule, when this molecule attached, the enzyme looks like this. Now you can say a smoothness in the site where enzyme is going to conduct the reaction. So this type of sites in enzyme, we call it a substrate binding site or active site of enzyme, where the substrate is bind. So if you see, for example, this is a substrate is not going to bind here because of the roughness or the site where the enzyme is active. Okay, so for activity of enzyme, it is necessary to attach its prosthetic group to the enzyme if it is a conjugated enzyme. Okay, so if it is a simple enzyme which does not require any prosthetic group or any cofactor or any coenzymes, in that condition enzyme can conduct a reaction without any support. Okay, so let's see a second condition. What is the prosthetic groups? Prosthetic groups are chemically bonded with the enzyme and necessary for activity of enzyme. Okay, so prosthetic groups are mostly chemically bonded, means there is a tight binding between enzyme and prosthetic group. So these prosthetic groups are non-proteinaceous. So let's see, there is one more thing which helps the enzyme for conducting the lecture, and that is a coenzyme. What are the coenzymes? These are the organic compound attached to the protein part of enzyme are called as coenzymes. For example, FAD, FMN, this is flavin, a mononucleotide. FAD, you know that? FAD. Let's see in respiration chapter. Okay, FAD, FMN, we are going to discuss this all type of organic compounds. So there is one more thing which help enzyme to conduct the reaction, and these are the cofactors. Okay, so let's see what are the cofactors. So these cofactors are the inorganic. Inorganic means there may be a Mg2 plus, calcium, Mn, Zn, ferrous. Okay, so zinc, manganese, magnesium, calcium, fluorine, sorry, iron. These are the inorganic part or the component which help enzyme to conduct the reaction. Okay, so these are the necessary factors. They just help enzyme to conduct its reaction, but these type of cofactors, if they are absent in the vicinity of enzyme will not work properly. Okay, so it is right, it is true that they are the accessory element, but it is necessary for the activity of enzyme. Okay, so guys, please understand this concept. Enzymes are the protein, but they require non-proteinaceous part. The first is your prosthetic group, we call them prosthetic group. It is organic compound, we call it a coenzyme. If it is inorganic, we call it a cofactor. Okay, and these elements are the important for the activity of enzyme. If any one of these is absent, then enzyme will not conduct its reaction properly, or the speed of enzyme will reduce drastically. Okay, so these are necessary for activity, but these are not enzymes. Okay, these are not enzymes. They required by enzymes for its proper activity. So terms which are used for enzyme are the first. The first term is apoenzymes. What are the apoenzymes? Apoenzyme is the name given to an inactive enzyme that lacks its coenzyme or cofactor. Okay, so let's see. Let's draw a diagram here. For example, this is our enzyme, we draw it before. Okay, so this enzyme is going to conduct a reaction where this is a substrate is a chemical on which enzyme is going to act, and here is our prosthetic group. Okay, or coenzyme. If this enzyme is without coenzyme, so now we don't have any coenzyme here, that time, or there is no any cofactor here. Okay, so only proteinaceous part is here, only protein is remaining. Okay, why only protein is remaining? Because there is absence of the coenzymes and cofactors, and at that point you can say this is a apoenzyme. Okay, so let's make a definition for apoenzyme. Apoenzyme is an enzyme without cofactor or coenzyme. Cofactor or coenzyme. So what are they? Apoenzymes are plain proteins. Okay, these are the plain proteins. Okay, nothing but the plain proteins. So what are the uh, holoenzymes? Holoenzymes is a term which is used to describe an enzyme that is complete with its coenzyme and cofactor. Okay, so now let's draw one more enzyme here. So this is the same enzyme I draw above. Okay, so this is same substrate what we are calling about, and these are the enzyme. So this enzyme now had its prosthetic group or its cofactor. Now you can say this is a holoenzyme, the complete one. Okay, so this is a holoenzyme. It is a complete enzyme. Okay, with its, so you can say holoenzyme is apoenzyme plus cofactor or coenzyme. Okay, so it is complete now. And this apoenzyme is what? Apoenzyme is holoenzyme minus cofactor or coenzyme. Okay, so if you get this term holoenzyme, then you can understand the what are the apoenzymes. Holoenzyme means complete enzyme which is with a proteinaceous part, and also with non-proteinaceous part, which is a active now. If you remove that coenzyme or cofactor, which is non-proteinaceous, that time the remaining portion of enzyme is called as apoenzyme. Okay, and holoenzyme means a complete enzyme. So what is holoprotein? It is the term which is used for the protein with a prosthetic group or cofactors. Okay, so for example, protein plus your coenzyme or cofactor, you can say holoproteins. Okay, and a coenzyme bind to a protein molecule, the apoenzyme. Protein molecules means apoenzyme. Okay, so when your coenzyme binds to a protein molecule, that is apoenzyme, to form an active enzyme, that is a holoenzyme. That means holoenzyme is your apoenzyme plus cofactor. Okay, that is what we written here. So this equation I write in this fourth bracket. Okay, so this is how our holoenzyme and apoenzymes used for the a single enzyme term. Okay, so if let's see guys, I am again explaining it. Apoenzyme means only protein part and holoenzyme means protein part plus its accessory element. Then whatever it is a cofactor or it is a prosthetic group or it is a coenzyme, it doesn't matter. But when enzyme is complete and it is ready to start or to catalyze its reaction, you can say it's a holo enzyme. Okay, so holo enzyme are always in active form and apoenzyme requires the factor which is necessary to activate its active form. Okay, so let's see properties of enzyme. What are the properties? The first property is a 3D configuration. Okay, so how this 3D configuration matters? Let's see, this is a substrate. Substrate means the molecule which is going to convert itself into the product, and enzyme is helping it to convert it into a very less time and less energy. So here is your enzyme which is acting over the substrate. So if you see the space for a substrate is exact in an enzyme. So inside the enzyme there is a exact space for a substrate. So this is a holoenzyme. Why? Because cofactor, your cofactor is here. So this is cofactor. So if you see this apoenzyme is a portion without cofactor and apoenzyme plus cofactor. Now you can say it is a holoenzyme. So this is holoenzyme with active site coenzymes and cofactor. Everything is there with a apoenzyme. So you can say it is a holoenzyme. In this enzyme, let's say this is enzyme and substrate is look like this. For example, this is another enzyme and the substrate of this enzyme is looks like this. So now you can understand the enzyme required exact 3D configuration. That's why its active site will be looks like a substrate, and then substrate binds to the active site to activate or to catalyze the reaction. Okay, so 3D configuration of enzyme or the shape and the site of interaction of enzyme is necessary to be in a exact configuration. Okay, so it matters guys. And when its site changes or when this 3D configuration changes, let's see if there is any change in an environment means there are few factors: temperature, pH, okay, or the substrate concentration. This factor if changes, then this protein which we call it apoenzyme will going to change its shape, and because of that there may be a uh, changes in activity or changes in activity of enzyme. It is why it is because of only a change in a 3D configuration. Okay, so it's a native structure. Okay, we can say a native structure which is necessary to conduct the reaction is important for the enzyme. If there is any change in its shape, there may be a change in its function. Okay, so here shape is important. for function of enzyme. Okay, that's why we call it a 3D configuration. 3D configuration is necessary. It is important to be maintained by enzyme. Otherwise, it will not work because its shape will get changed. For example, this enzyme is now out of its 3D configuration. It will looks like a, and the substrate is here. So you now distinguish between the 3D configuration and the enzyme which is not in a 3D configuration. Okay, so here substrate is matching the site or active site where it is going to bind, but this substrate is not uh, compatible with the enzyme because enzyme get change its shape because of the any reason. Okay, so there may be a change in temperature, change in pH, or change in a concentration that is hampering the structure of the enzyme. What is catalytic activity? Catalytic activity means without changing the position of equilibrium, enzyme can accelerate the rate of reaction. Okay, so catalytic activity means enzyme. What is catalyst? Catalyst speed up the reaction. Speed up the reaction. So catalysts are mostly used to speed up the reactions. So which reaction? Any reaction where enzyme can participate, it will definitely speed up the reaction by changing in its activation energy. But what is meant by change? If you see, this is uncatalyzed reaction. Okay, so here is the first reaction in the first graph which is without catalyst. So if you see, the activation energy of a reaction is more if you add an enzyme as a catalyst. So the energy of activation from here, it reduces to the here. So when enzyme is involved in a catalysis of reaction, that time it requires a less energy, and because of that I said without changing the position of equilibrium, enzyme catalyze the reaction by minimize the activation energy, or the speed up the reaction by minimize the activation energy, but without making any change in a position of equilibrium. Okay, so position of equilibrium is not going to change at any cost, my dear friends. Okay, so it's very important to keep in mind, enzyme reduces activation energy, enzyme minimize the activation energy. Okay, so let's see, enzymes are the biocatalysts that increase the rate of reaction without itself being changed. That means when this reaction is conducted by enzyme, so this is your enzyme, when it conduct this reaction, the substrate will be like, okay, so enzyme conduct reaction, so it converts its one substrate into a two product, and after all the product will be a move to the function or the next reaction if it is present. Otherwise the enzyme will come and it is now ready to function for a same reaction with a next substrate. Okay, so this is how enzyme catalyze the reaction. A single enzyme can catalyze multiple reactions, but the substrate is fixed. Okay, for example, you are taking the enzyme of cellulase. Okay, cellulase means enzyme which can digest the cellulose. It can, a single enzyme can digest multiple bonds in the cellulose, but it will not going to digest any bond from the starch or the other molecule. Okay, so the substrate of enzyme is a fix, but if you add more number of same substrate for the specific enzyme, it will conduct multiple reactions one by one. Okay, multiple reactions means not simultaneously. It will conduct one by one. And how many reaction are depending upon the enzyme. Okay, crores of reaction can be conducted by a single enzymes. So after the catalytic activity, we know that this substrate is going to convert into the two product, and because of that, the enzyme intact or integrity of enzyme will not lost, and it will come again to take part in a new reaction. Okay, what is the specificity? This is a best property of enzyme. If you see the specificity of enzyme, uh, enzyme is specific to the substrate. What it mean? It mean, let's see this first diagram. This first diagram explains the size of a substrate, and the site where it is going to bind is looks like a similar, or they are compatible to each other. This substrate is also going to bind here properly. Okay, and this substrate is also going to bind here properly. So this is a specificity. That means an enzyme can carry a multiple reaction, but this enzyme each and every time carries a reaction with the same substrate. It will not move to this type of substrate or the another substrate which looks like a different from its active side. Okay, so this is a specificity of enzyme. One enzyme can conduct multiple reaction, but the substrate will be a same for that particular enzyme. So this type of property of enzyme, we call it a specificity of enzyme. Okay, let's see factors which are affecting the enzymes. So there are a few factors which are included in our syllabus. The first one is the temperature, second one is a pH, and third one is a substrate concentration. Okay, if you see the temperature, enzyme shows a maximum activity at this temperature, which is very close to the body temperature. Okay, so this is for human, I mean this is for the organism which are working, or the body temperature of that organism is near about to 37. But if I take enzyme or the organism from hot spring. ओके, हॉट स्प्रिंग म्हणजे काय रे मित्रांनो, गरम पाण्याचा जो झरा आहे त्याला आपण हॉट स्प्रिंग म्हणतो. इन दॅट हॉट स्प्रिंग द टेम्परेचर ऑफ वॉटर इज मोर दॅन 70, 80 and 90 also. Okay, so at that time the organism which are living in this water, we call it a thermophilus. Okay, so philic means loving and thermo means heat. So organism which are loving to be a part of warm environment. Okay, so in that condition this organism or the bacteria, let's take an example, bacteria. Okay, so this bacteria had enzyme which are working on the 70 degrees Celsius. Okay, because it is living in 70 degrees Celsius. Okay, so this is how the optimum temperature of enzyme will change with respect to the condition and with respect to the organism and with respect to the environment where it is living. Okay, so you can say, for example, in your body there is a gradient in a temperature. For example, brain is working on different temperature. It is not right, I am just taking a condition. Okay, just consider it, brain is working on 10 degrees Celsius, your liver is working on 30 degrees Celsius. Okay, so that time your enzymes which are working in a brain will never activate in the liver, and the which are working in a 30 degrees Celsius of liver will never conduct any reaction into the brain where the temperature is 10 degrees Celsius. Okay, it is not right, guys, it is just example we are taking to our concentration. Okay, so let's see the pH. If pH changes, it will again change the rate of reaction. Okay, so let's see what is optimum pH here. Optimum means the value of a temperature. Optimum temperature means, here I write optimum pH, the value of pH where enzyme shows a maximum activity. Optimum means where maximum activity enzyme shows. Okay, so optimum pH means a value of pH where enzyme shows maximum activity. So let's see where is maximum activity of enzyme. This is a pic of our graph. So let's see, At that point, I am going to draw a line on the pH scale. Okay, so here we know that it's about a 7 to 8 pH. So in this pH, enzyme shows a maximum activity. Okay, again it is for the enzymes. We are conducting the reaction into the neutral pH. इट्स अ न्यूट्रल 7च्या जवळपास आहे म्हणजे न्यूट्रल म्हणू शकता तुम्ही त्याला बरोबर की नाही सो दिस आय एम टॉकिंग अबाउट दी ओनली enzymes which are going to conduct its reaction in a neutral pH. But if you see enzyme which is a pepsin, part of our stomach, pepsin is present in our stomach, it will work uh, with its maximum rate at 2 pH. Okay, so at what pH? At 2 pH, it will work with its maximum rate, and because of that, we call it the 2 pH is optimum pH for the pepsin. Okay, so 9.5, it is a trypsin. Trypsin is a part of our duodenum, or we can say small intestine, where